So without further ado, I would request Professor Jeff Darabi to.
I'm excited to share with you some of the work that we have done on microfluidic devices for cell and particle separation. I'd like to acknowledge that I am here today because of the work, all the work done by my students. I will also acknowledge at the end. So in my lab, I mainly work on two main areas. One is on basically microfluidics for cell separation, which I'm going to talk about today. And the other area, which is actually I work more, is on thermal management of high heat flux electronic systems. But my talk today will be on bioseparation chips. So I'll talk about mainly two techniques, magnetophoretic-based cell separation techniques and dielectrophoretic-based cell separation techniques. So what is magnetic bioseparation? So this technique has been around for decades. Mostly biologists have used it. So it's a separation technique that you can use to separate DNA, RNA, bacteria, cells from complex mixtures such as blood, saliva, other samples. So as you can see in this slide, normally you will need, because cells are not magnetic, you need to first tag it with magnetic beads. And for that, you need an antibody that is specific to the cell or particle that you want to separate. So these kits are commercially available. They are pretty expensive, but you can buy them. And in conventional tube-based systems, as you can see again in this cartoon, let's say, for example, in this case, your target cell is the green. And you want to separate this for a mixture of other cells. So you mix your sample with beads and antibodies, and then after incubation, you can place them in a magnet, which looks like this. So what happens is that once you place a sample in a magnet, the target cells, which have beads attached to them, they are retained on the inner wall of the tube. And then you can remove the supernatant and resuspend the target cell. So it's fairly straightforward. However, there are a lot of limitations with the conventional tube-based systems. For example, you have significant cell loss due to multiple washing, pipetting, transfer, et cetera. And also, this technique is very time-consuming and user-dependent. So what we have done, we have developed a continuous flow microfluidic chip that we enter, like the sample is introduced into the chip. So again, we have alternating polarity. We use an array of like a small magnets to create what we call the long-range magnetic field gradient. In addition, we deposit and pattern a thin film, nickel grid, to create, again, what we call short-range magnetic field gradients, which I'm going to explain later on in the modeling part. So what happens is that this is the nickel grid, and as you can see, we get a uniform particle trapping at the edges of this nickel grid, which significantly improve the purity of the isolated particles. Now, there are, you heard from other talks that there are advantages with microfluidic-based bioseparation devices. So the reagents that are used for these applications are very expensive. To give you an idea, when you buy a kit that you can like separate maybe 50 samples, it costs about $1,000. So, but when you use like a microfluidic-based devices, you can save a lot of reagents. They have, we'll show later on that we get a better performance and perhaps the most important thing is the integration and automation that cannot be done with batch separation techniques. Some of the applications include basically isolating cells. That's one of the main applications. They are used for stem cell research. They are used for like pathogen detection in water and milk, etc., as well as to separate DNA and RNA. We have come up with a relatively simple fabrication technique. Basically, we start with a glass substrate. We deposit and pattern a thin film of nickel. Then we use a commercial plotter to cut the fluidic channels in a double-sided Kapton tape, which is shown here. This is basically this is the top view of the polyamide, or Kapton tape is the commercial name. Then we sandwich everything. Basically, we also make the top cover with the inlet and outlet ports. Then under pressure and heat, we assemble the device. So like my students, after they become skillful, they can make this device in 2 hours. Which saved, initially we had to make the channels in glass by etching, which is very time consuming and expensive. So this is a schematic, is photos of the experimental setup. We use 2 pumps. Oh, I'm sorry. So we use a bidirectional pump for the sample and a syringe pump for the buffer. This is a photo of the assembled device under basically microscope for testing. We initially tested our device to separate CD4 positive cells from human blood. There is a special kit for this, commercially available. There is a standard protocol how to separate mononuclear cells from blood, which is shown here. Then after separation, we do the flow cytometry analysis to quantify the performance of our device. So this slide shows the, this is called basically dot plot. I work with a biologist that has a flow cytometer that does the basically flow cytometry analysis of the samples. So this shows a sample which had 17.2% CD4 cells. So I should mention that CD4 positive cells are identified in this quadrant. And then Q1, Q3, and Q4 represent non-target cells. And then as you can see, after isolation, most of these non-target particle or cells are gone, and we were able to increase the purity of the sample from 17% to nearly 97% in a single step, which is considered very good compared to the conventional techniques. We tested our chip at different flow rates. Our target is to separate 10 milliliters in 10 minutes, which is considered actually quite high for microfluidic-based devices. So you can see here that the purity of the sample increases with increasing the flow rate. However, the recovery, which is the ratio of the isolated cells in the to the total number of target cells in the initial sample, slightly decreases. So there's a trade-off between purity and recovery at that higher flow rate. Now, after successful isolation of CD4 positive cells, then we tested our device to separate cancer cells. This was our primary target. Again, there were several talks about circulating cancer cells. So what studies have shown that cancer cells are shed from the primary organ and enter the bloodstream. So if you can come up with a device that we can capture and detect these cancer cells at early stages of the disease, that can help with a early treatment and or diagnosis of the disease. First, we did some basically cancer cell bead coupling to make sure that the beads attach to the cancer cell. And then after successful bead CTC coupling, so here you see these are the beads, and these are the basically cancer cells. Then we tested, my collaborator, so he has a cell culture facility, cultured MCF7 cancer cells. We spiked them into the sample and then try to see if our device can isolate these cells. So again, there was a talk about, these are like a lot of biologies involved. CTC cancer cells have to be basically positive by DAPI, which is a stain to identify a nucleus, as well as also a secondary antibody that identify the cancer. And this shows a merged image that identifies that this cell that is separated is actually a cancer cell. Now again, after showing cancer cell isolation, we did the test with by spiking cancer cells in diluted blood. And again, as you can see here, we were able to capture the cancer cells. So these are done at extremely low concentrations, around like 50 cancer cells per milliliter. Studies have shown that the concentrations of cancer cells at early stages of disease are actually like a handful, like less than 10 per milliliter. So the challenge is your device has to be able to capture those very low concentration of CTCs. So we did the recovery test, and as you can see here, we were able to capture or recover nearly 80% of the cancer cells that we spiked into the blood sample. So we also, again, as I mentioned, most of these studies traditionally have been done by biologists, which they don't like math. So we have done a lot of work on modeling. We use OpenFOAM. For those who are familiar, is an open source software program. A little bit hard to learn, but is more flexible. So we simulated the magnetic field, fluid field, and particle tracing. Again, I've summarized some of the equations here given by these equations, but in the interest of time, I'm not going to get into details. So as I mentioned earlier, we use an an array of magnets with opposing polarity. So the reason for this is that to create these peaks, which basically have much higher magnetic flux density. In addition, so the nickel gray that I showed you earlier, we also get high, what we call short range magnetic field gradient at the edges of the nickel grid. So this helps us to get a better performance compared to conventional techniques. So this is the animation of our computational modeling. So here you'll see 4 windows taken along the channel at different distances from the inlet. So in the entrance region, so the particles are randomly distributed. And then you have so several basically cases. We have cells with 20 beads, 10 beads, and 5 beads, 2 beads, etc. So this window shows the trapping region, roughly 11 millimeters from the entrance. As you can see, so the red cells, which have 20 beads attached to them, are captured first, followed by the yellow, which have basically roughly 10 beads. Then we have this light blue. They get captured between 17 to 20 millimeters from the inlet. And you'll see that cells which have only 2 beads attached to them, so the magnetic force is not large enough to pull down the cells and they flow through the channel. So, but we can play with the different parameters to optimize basically our device for different applications. So then we did some parametric studies to study the effect of different parameters, including, so this one shows the effect of beads, which is the same as the earlier animation that you saw. So as you can see, as the number of beads decreases, then the trapping length increases, and in some cases, like in this case, they even do not get captured and then flow through the channel to the outlet collection. We have studied the effect of the basically cell diameter. So here you can see that as the diameter, again, when we study one parameter, we keep other parameters constant. So in this case, the number of beads are constant. So as you can see again, for larger cells, so 10 beads is not sufficient, and then the trapping length increases with increasing the cell diameter. Another parameter that is important is the flow rate. So flow rate, on one hand, we prefer higher flow rates because that makes the separation faster. But on the other hand, if the sample flows too fast, then the magnetic force may not be sufficient to pull the particles down and capture them. So here again, we keep the number of beads and constant. Then as you can see, as the flow rate increases, these are like a very large flow rates. So at certain flow rates, again, the force is not enough to pull the cells, to capture the cells. Now I'm going to switch gears, and so the advantages of the magnetophoresis by particle separation is that so the throughput is quite high. So as you can see, we have successfully shown flow rates up to 60 milliliters per hour. Now, the drawback is that for some because once you done with the isolation, then you have antibodies and beads attached to your cell. So there is a reagent that you can use to cut the antibody and then separate the isolated cells. But for some applications, such as like gene expression, those impurities are not acceptable. So then you need to have like a pure cells. So in that case, then we could use dielectrophoretic bioseparation techniques. However, with this technique, as you'll see later on, the throughput is very small compared to magnetic particle separation. So again, the DEP force is given by this equation. As you can see, the force is proportional to the volume or radius A cubed, where this FCM is called Clausius-Massati factor. Depends on the complex permittivity of the medium and the particle. So again, if we have a particle in a uniform electric field, the particle polarizes. It experiences equal and opposite forces in the both directions, and there is no translation. So we will need a non-uniform electric field. Again, if you have a neutral particle, it polarizes. And but since the electric field is different at both ends, then we get a larger force on one side, and this is enough to move the particle to the one side depending on the relative permittivity of the medium and the particle, which are referred to as positive dielectrophoresis, is where the particle moves towards the regions of high electric field. And in some situations, so the particle actually moves towards the regions of low electric field. So we can use this to, again, separate particles. So in our approach, what we have done, we are using slanted electrodes on top and bottom substrate, as shown here. So what happens, we are using negative DEP force, which repels the particles from the regions of high electric field. In this case, they are basically repelled from top and bottom of the channel and towards the center line. So they line up through the center line, and then we have a lateral DEP force that deflect the particle in the Z direction, in lateral direction. So we can separate the particles based on their size and direct them into different channels. So obviously, so here the larger particles experience a larger force and they are deflected more. Again, I will show you some animation later on. Again, for this one, we have done the modeling in open foam. So this one requires, again, the electric field simulations, the fluid field simulation, as well as the particle tracing. So this is the computational domain. These are the electrodes. So the electrodes in this case, they are 20 microns wide and with a gap of 20 microns. Again, the governing equations are very similar, only the DEP force is different compared to the previous case. So this shows the electric field simulations. Again, we get these spikes at the edges of the electrodes. So that's what's called like fringing effect. And as different distances from the electrodes, so as you can see, as the distance from the electrode increases, the electric field kind of levels off. Now, this slide shows the DEP force in X, Y, and Z direction. as I explained earlier, so because of we have like electrodes on top and bottom substrates, so they repel the particles to the center line, so the DEP force in the normal direction is 0. So you can see that's the green one. And then we are using the Z component, so which is the blue, to deflect the particles in the lateral direction. So this animation just showed 2 particles. So here the force is so strong that the larger particle basically, once it reaches the first electrode, is deflected, moves along the electrode, and eventually turns and goes to the separation channel. But we did a large number of particles. So here we have 3 particles. Again, depending on their size. So the yellow just moves along the first electrode and deflected, and then you see what was the green one, somewhere in between, and then followed by this cyan or light blue. So again, we had to optimize our device by studying the effect of different parameters, including some the particle size, voltage, flow rate, channel height, etc. So I'm going to share with you some of the results. So this slide shows the effect of the particle size. Again, we keep all other parameters the same. Just so as you can see, the larger the particle, the larger the deflection, as expected. So this slide shows the effect of the voltage. Again, if we increase the voltage, but while keeping other parameters constant, the DEP force is larger, and then we get a larger deflection. So this shows the effect of the flow rate. Again, so in this case, so the higher the flow rate, actually the smaller the deflection, again, because the force is not large enough to deflect the DEP force is not large enough to deflect the particle. The channel height, so in this case, if we increase the channel height but keep the voltage constant, again, so obviously the DEP force would be a smaller, therefore a smaller deflection. We studied the effect of the number of electrode pairs that we put on the substrate. So again, you can see the effect of the number of electrodes. So the larger the number of electrodes, the higher the deflection. I think that's it. So to summarize, we have successfully demonstrated basically isolating cells using magnetofluidic and dielectrophoretic-based microfluidic devices. We have, I think, among the first who have done extensive computational modeling of these systems. Now, even though the performance that I showed you are quite good, so our goal is to have like someday a point-of-care diagnostic device. Right now, once we separate our the target cells, my students spend like 2 hours under a microscope to scan and count these cells, which is not acceptable. So I'm in discussion with right now I have a let me actually go to the collaborating with Professor Joseph Schuber, who does the biological part, and in the process of teaming up with a faculty from the computer science department with the computer vision background to try to train and automate the cell counting after the separation. So that's our next step. So I'd like to acknowledge my students who have been working on these the studies that I presented and the funding agencies. Thank you for your attention, and I'll be happy to entertain any questions that you may have.
So thank you, Professor Darabi, for an interesting talk. So it is really interesting to see how external fields can be used to manipulate or sort and separate bioparticles. So now the floor is open to questions.
Yeah, so very nice microfluidic device. I didn't quite understand. You have nickel on the surface,
Yeah.
and you have them oppositely magnetized. How
Right.
can you pattern it with nickel, and then you have
what
north to south next to each
So
other?
basically, we use those magnets that we commercially purchase, they have a thickness of like 1.8 millimeter. So what we found, because of the high gradient at those alternating point, we got some clustering. So by depositing the nickel, so then when the cells come down, then we basically the external magnets energize the nickel grid. So then this, as you saw in the image, then the particles kind of like uniformly line up along the nickel lines, which has a higher magnetic field gradient as well.
And do you have a number for the gradient in Tesla per meter or something? Like
I think what was in the, it's about 0.2 Tesla per meter, yeah.
oh, okay.
Yeah,
At the level of the cells.
yeah.
Is there a limiting factor to the particle size so that the dielectrophoretic effect is dominant enough for particle sorting? Means, have you worked with particles in sub-micron range?
So actually, we have done a very, very good question. So when we go to sub-micron range, now that I have, I didn't want to basically present, but we have done like for DNA and RNA, they are sub-microns. So in that case, the Brownian motion would be important. So we have recently published a paper on that. I didn't discuss it. So so here. So when we have like sub-micron particles, then this is like 500 nanometer. So then the Brownian motion, basically the green line shows, again, the trapping distance without Brownian motion. But at that scale, the Brownian motion becomes important. Then as you can see, you have some random motion of the particle, and that will cause the trapping region to have actually to have a bond. Is not a single point as we discussed for the larger cells. So that's a very good question. We have done that, but I thought it would be too much to present today. Yeah.
Thank you.
So that's why the cancer cells are not magnetic.
Yeah, so
that's why you have to basically tag magnetic beads. There are like specific antibodies that on one end attach to the cell, and on the other end to the bead.
Yeah, yeah.
So again, this is a standard protocol that has been developed by biologists and chemists. That's beyond my actually expertise, but they are commercially available. So
it was the bead was attached to it before you started using it? Or
no, you buy you buy antibodies as well as beads, basically separately. That's why before doing the experiment, we had to kind of optimize the ratio of the antibodies and beads to make sure that we tag enough beads to the cancer cells.
Here.
Yeah, sure.
So thanks for a nice talk. So I'm intrigued by the data you had on the CTC separations, and maybe I was not paying attention, but you mentioned you're using diluted blood.
Yeah.
So how much is that dilution?
Like 10 to 1. So 10 to 1. Like
10 to 1?
Yeah, like actually 1 to 10. Yeah, we
Okay.
dilute 10 times.
Yeah, okay, but that's diluted whole blood.
Yes.
From
Yeah.
sheep. Those were the sheep blood.
So we initially basically tested like we did this like in the steps. Initially we added cancer cells to the red blood cells, like,
yes,
like sheep red blood cells.
Yes.
And then we used basically human blood, but diluted human blood. So
okay, so when you did human blood, it was diluted 1 to 10.
10, that's correct.
Okay, and
so like that may not be like we like to, if you go to like a whole blood, that would be a kind of like a different story. Then the modeling part has to be, you go to the non-Newtonian fluid, but also is working with the whole blood is very difficult. So,
Yes, indeed.
yeah, so we have to basically, with 10 times we are not happy, but we can probably go to maybe 1 to 5.
Okay.
So that's still
And
ongoing.
the sample throughput was, I didn't follow that.
So the sample throughput we have done, like the results that I showed you for recovery, roughly 82%, is for the 60 milliliter per hour. So and then
So 1 mil per minute, yeah.
yes, and
Yeah,
then
okay.
so that's basically we can separate even if we dilute, let's say 1 milliliter blood 10 times will be 10 milliliter.
Yeah.
So we can separate that in 10 minutes, which is I think is reasonable.
Yeah.
So for clinical applications, which I mean time is important.
Yeah, and
so.
you need like a full tube, so 7 mils or so to do but that's still within an hour or so. Okay, thank you.
If you do basically with a dilution, that's correct.
If there are no more questions, then let's thank the speaker once again. We would like to offer a token of appreciation to Professor Darabi.
All right, thank you. We can just shake hands.
The next